Historical to cutting-edge progressions in hematology. Ancient foundations. Hematology origins lie in ancient observations of blood as a life force. The Greeks, like Hippocrates, linking blood to health via hematology without scientific precisions. Bloodletting was accordingly early interventions. 17th-19th century. The microscopy's invention 17th century allowed Antonia van Leeuwenhoek to describe red blood cells. In the 19th century, William Addison and Gabriel Andrel identified white blood cells, laying groundwork for understanding blood compositions. Blood transfusion attempts begin often with disastrous results due to unknown blood types. Early 20th century. Carl Landrin's discoveries of bio blood groups. 1901. Revolutionized transfusions. The hematocrit and hemoglobin museums emerged, enabling anemia diagnosis. Bone marrow role in blood cells production while clarified, and pernicious anemia was linked to vitamin B12 deficiency. Mid-20th century. World separate advances in blood banking and plasma fractionations. The CAMPS test improved hemolytic anemia diagnosis. Sickle cell anemia was characterized as a molecular disease. Linus Bolling, 1949, marking hematology's molecular turn. Chemotherapy for leukemia and lymphoma began with methotrexate showing promise. Late 20th century, bone marrow transplantations between 1960s became a game changer for hematologic malignancies. The discovery of hematopoietic stem cells and growth factor, e.g. erythropoietin, depends on understanding of blood productions. Molecular genetic dependent oncogenes, e.g. BCR, ABL in chronic myeloid leukemia, enabling targeted therapies like imatinib 2001. 21st century. Hematology now embraced precision medicine, gene therapies, and immunotherapy. CRISPR-based treatments for sickle cell disease and thalassemia are clinical use. CAR T-cell therapies transform lymphoma and leukemia outcomes. Artificial intelligence aids diagnostics predictions outcomes from blood smears or genomic data. Liquid biopsies detect minimal residual disease, guiding therapy. Meanwhile, global health focuses on equitable access to the advances. Lecture 1. Gene editing for hemoglobinopathies. Title: Writing the Code of Life. Gene editing for sickle cell disease and thalassemia. Historical context: sickle cell disease, SCD, and thalassemia, inherited hemoglobinopathies, have challenged medicine for countries. SCD's molecular basis was elucidated in 1949, revealing a single amino acid mutations in hemoglobin. Thalassemia's genetic defects were mapped in the 1970s. Treatments like transfusions and hydroxyurea improved outcomes but didn't cure. Bone marrow transplants introduced in the 1780s offered a cure but were limited by donor availability and risks. Current advancements: gene editing, particularly CRISPR-Cas9, has transformed the field. In 2023, the FDA approved Casgevy hexagonal autotemicel, a CRISPR-based therapy for SCD and transfusion-dependent beta thalassemia. The process involved editing hematopoietic stem cells to post-fetal hemoglobin (HbF) expressions, compensations for defective adult hemoglobin. Clinical trials shows patients achieving transfusions independence and symptom relief. Base editing, a more CRISPR variant, is tries targeting specific BCL11A innocence mutations to minimize off-target effects. Lentiviral gene therapies like Siontigio for thalassemia also show promise but are considered. Challenges and future direction. I costs between 2,002 million per patient's limit. Access, especially in lower source setting where SCD is prevalent. Long-term safety data are incomplete. Call editing cells lead to malinosis. Scalable delivery models and in vivo additions directly in the body are under explorations. Equity in access in virtually as most patients live in sub-Saharan Africa and India. Research also eyes gene editing for other hemoglobinopathies like alpha thalassemia. Takeaway: gene editing offers a functional cure of hemoglobinopathies, but its promise hangs on affordability and global reach.